Hello everyone, I'm Katie. I'm a recently graduated PhD from Daryl's lab, and I'm going to tell you today about CRISPR design. And basically, one of the main things I want you to take away is this is just how our group designs and works with CRISPRs. There's a lot of different ways. Using CRISPRs is basically just a technique to do the gene editing that you're interested in doing. So I'm going to explain to you how we do it and think about it. Oh, sure. Thank you. Cool. But of course, there's lots of different approaches, and I would love, this is sort of general because obviously I don't know exactly what you all are interested in, but so feel free to ask whatever questions. And I also have no idea what Gustavo told you, so I apologize if it's a little bit of overlap. I missed that part. Okay, so again, I'm going to tell you about CRISPR-mediated gene editing. And the general idea, again, is that the CRISPR-Cas9 system is a tool that you can use to make a very precise nuclease-induced double-stranded break in DNA. And this allows for you to do a number of possible repairs. So you can basically, by not giving the DNA a template to repair, you can hope that it might make a deletion or an insertion sort of at random, which can cause a gene or regulatory region knockout. What I'm mostly going to talk about and what I have experience with is using a donor template, which means that after you make your double-stranded break, you then tell the cell and the DNA what sequence you actually want it to make, and then some percentage of the time it will read from your donor template, and you get homology-directed repair and an insertion or a modification that you're telling the DNA. So, right. Okay, so just to give you a sense of the workflow I'm going to go through for CRISPR gene editing. There's a number of different steps, and again, depending on your interests or what you're trying to do, you might be able to skip or modify any number of these steps. So the first thing we're going to talk about is how to do in silico design of guide RNAs. So the guide RNA is going to tell the Cas9 where to cut, so this is an important process where you design an appropriate targeting strategy, basically. I'm going to tell you about how our lab generates the reagents to introduce the guide RNA and the Cas9, as well as the donor template if you're using it, to the cells. And then talk a little bit about how, like, some options for how you might test these constructs, transfect them specifically into pluripotent stem cell lines, screen and clone out the colonies you're interested in. So the first, again, as I said, is just going to talk about how you in silico design guide RNAs. Also, I don't know if I said this, but you should interrupt me whenever with questions. It will make me feel better. All right, so the first question then is that you basically have to answer for yourself, depending on the experiment or the question of interest, is where do you want to target your guide RNA? And whether you want to do a knockout or an editing, as I said before, you need to design a strategy where you target the region that you want to edit. So really the important thing we're going to talk about now is this idea. So here, you might, for a knockout, you might design a guide RNA that targets, for example, an essential domain of your protein, so that when it cuts and the gene is repaired wrong, so it deletes a region, for example, that region is something that's important to the function of the protein. If you want to make a specific edit, you might have to be a little bit more precise with where you target. So a lot of what I'm going to talk about is generating reporter lines for a specific gene locus, and in that case we typically target the stop codon of the gene of interest, so that when the cell makes that particular protein, it also makes our, our reporter line. And again, this will depend a lot, and I guess I would emphasize is just a really important point to make sure you think about a lot where you want to target your CRISPR. Because once you've decided, actually designing the guide constructs is super easy. So don't know exactly how you guys want to do this. Basically, I was just going to go through a potentially kind of boring explanation of the specific steps of how you design a guide RNA for a gene of interest or a region of interest, and then maybe we can have some time where I can like show you on my computer how these steps actually look, or I know some of you brought your laptops, so if people have specific questions, we can go over them then. So what I would do is, once you've decided, okay, I want to target, for example, the stop codon of this particular gene, you just select 250 base pairs of the sequence around that site that you're interested in. The best way to do this is to use the NCBI website to find the exact sequence of your gene. You can basically download an annotated transcript of the locus and put it into any sort of any sort of software that can tell you about DNA, like annotations of DNA. So we use Snap Gene Viewer, which is free software, and also has a full version you can pay for that's really useful for cloning. Again, that's just what we use, and I think there's lots of other software depending on what your labs are interested in. But anything where you can basically download the sequence and then it will be fully annotated, so it will tell you everything about where the exons are, what the coding sequence is, and things like that. And again, we can look at an example of that in a minute. So using all of that information, you just identify the DNA sequence of the region of interest and select those base pairs. You then paste the sequence into an online tool. Again, this is all very easy. Basically, you just paste the sequence into an online tool. This tool I've actually never used. I used to use the tool from the Zhang lab at MIT, but they've recently said that their tool is actually outdated, and this is the one they've been recommending from a recent review that sort of tried to look at all of the options. And basically, this is going to give you a whole list of all of the sequences that meet the criteria that you need to be a guide RNA. And the main criteria depend on the Cas9 protein that you use, but in particular, and using this tool, you can tell it what Cas9 protein, but the idea is that you find 21 base pair sequences, and with the most common Cas9, they'll have a PAM sequence that's an NGG. And then it will look throughout the genome for all the other sites that that guide RNA sequence might bind to, and it will basically make you a list of how good it is based on its predicted binding and its off-target effects. And then with that, you select your candidates. It's good to select at least 3. You can try to select sequences that, again, minimize the off-target effects while being close to the site you want to target. And you can always double-check your guide against the exact sequence of the line you want to target in case of SNPs. And this tool that I pointed out will actually tell you if there's like common variants in the genome in the region you're looking at. And then you basically just, once you pick your CRISPRs, you can add them to your file that you've been working with in SnapGene or whatever program you use, so you can keep track of them. You can also use Microsoft Word, which I used to do, but don't necessarily recommend. Anyway, so I know that's just like sort of terrible slides with a lot of text, but I printed them out for you so that you can have a copy, so it's more like a little very vague protocol. Okay, so once you get through that sort of pretty straightforward step, the really important thing is to also design, so in addition you have to design a donor template if you are interested in using a donor. And again, a donor is useful if you want to, for example, introduce a specific mutation. If you want to correct, for example, a genetic mutation that causes a disease, you might give it a donor template that has the normal sequence. And I use donor templates, which I'm going to describe to you, to make reporter lines. So I already went over this, but you might not need a donor if you just want to make a knockout, and people have had success in our group with doing this, or a specific edit, you design a donor. So the requirements for designing a donor construct are pretty specific. Basically, what you're giving the cell is a repair template. So you want to design a template that's going to be entirely integrated into the genome and that has all the features that you're interested in. So with that in mind, it can be useful to include in your donor an antibiotic selection cassette so that when you target the gene or the DNA, if your donor is actually integrated, you can select using antibiotic selection only the clones that were targeted. You want to make sure to include a homologous sequence that's not to be changed at both the 5' and the 3' ends of the desired site that you want to edit. And this is so that the donor and the DNA that's being repaired knows that that's supposed to fit in there. So you want it to be able to go to the DNA that's damaged and bind to the sequences around the cut site, but then have this new template that's able to repair. You want to make sure that your donor can't be cut by your Cas9. So because you're targeting the same sequence, it would be bad if you had an identical sequence in the Cas9 site that gets snipped and then your donor doesn't work. And so you can either remove or make a silent mutation depending on the location of your cut site. And then you can either, depending on the length of the sequence you want to edit, have an oligonucleotide versus a plasmid donor template are sort of the main methods that our group ever uses. So for example, if you wanted to edit just like a single base pair mutation, you might only need like a long oligonucleotide because you don't need to insert very many base pairs. On the other hand, if you want to make a reporter, you might have a whole plasmid because your construct is pretty long. So that probably all was very vague. So here's an example of a donor construct that I use. So this gene is a gene called STGB3A2 that's expressed in the airway. And this is the native gene sequence. And we have a CRISPR that cuts right near the stop codon of the gene. So then the donor template is here. So it has this arm of homology to the 3rd exon of the gene, and then it has a 2A peptide and the reporter sequence, and then an antibiotic resistance cassette and another homology arm, which you can sort of see indicated by these arms here. So what this means is that if I cut using the CRISPR at this site, some of the time when this cut is repaired, it's going to read off of this donor construct. And instead of just going from exon 3 into the stop codon into the 3' UTR, it's going to add in this tag BFP, fluorescent protein, and this antibiotic resistance cassette. And then, yes?
So is there an upper limit to what you can insert?
I
guess there is, but what
yes.
sort of
Yeah, so I guess I would say the upper limit is, in my experience, mostly determined basically by how big of a plasmid you can clone. I don't know in terms of like if you could make a giant plasmid, if it would be integrated. This is probably, it's pretty long though. I mean, this is several kb probably, yeah. So, but I know some people in our group have had issues where they want to put in like an extremely long sequence and it's basically just impossible to clone. Are there any more questions? I know this is all kind of fast and like broad, but cool. Okay, so once you've decided the donor and the guide RNAs and everything you want to use, and this is all basically done on a computer, you have to actually make them. So that's the next step. So the first thing you have to do is decide, well, how do you want to get your guide RNA and your Cas9 into your cells? And what we typically use is a plasmid that expresses both a Cas9 and the guide RNA from the same plasmid. And this is nice. It improves the efficiency of the system, right? If you have to put in the Cas9 and the guide separately, you have 2 plasmids that need to get in. If you put them in together, you guarantee that any cell has both, which is useful. So we use these vectors from the Zhang lab that you can get on AdGene. There's a number of different flavors, but you can get them in particular in puromycin, which means that you can screen using antibiotics for the cells that got your plasmid, or you can also get ones that have GFP, which again means that you can sort out the cells that get the plasmid. And this is really useful because your efficiency is probably going to be pretty low. So you want a way that you can select out only the cells that got your guide and your Cas9. And it's really, the nice thing about this system is that their group designed it to be super, super easy to use. So it's basically very simple to, when you have your designed guide RNAs, insert them into this plasmid using just a simple restriction digest, basically. So this is the plasmid with this insertion site with these BBS1 sites. And basically all you do is you follow their directions, which are online, for how to order 2 oligos. You anneal them to each other. This blue here is your guide RNA sequence that you picked. And you basically just have these 2 oligos, you anneal them to each other, and then you cut this plasmid with these BBS1 enzyme, and you insert this guide using just like a simple ligation. So obviously I'm sure there's like a variety of cloning experience here, but in general, it's very easy. If you can clone anything, you can clone a guide RNA into a plasmid. And again, all the instructions are online, but of course, if you had specific questions, I could definitely answer them. And with that in mind, again, this is not a great slide, but I thought it would be useful information. There's this whole website where they explain all of these reagents and basically give you like a super straightforward protocol on how you use them. And they also, this same group has a Nature Protocols paper that's pretty straightforward. And AdGene also has a whole section about CRISPRs. So this step is actually pretty straightforward. And really the important thing is deciding the sequences you want to insert. Cool. Okay, so now maybe more relevant to this course is, so you have a guide RNA, maybe you have a donor, but now the important thing is actually targeting an iPS line with these constructs. And so you can test the constructs in advance, you can transfect them directly into your iPS line, which is what I normally recommend. Then you want to perform antibiotic selection if possible, and I'm going to talk also a little bit about single cell sorting, and then clonally expand and screen for successful targeting. So there's kind of a divided thought on whether it's like worth testing your CRISPRs before just like going ahead and using them in iPS lines. I personally don't think it's that useful because I feel like a lot of times they work really well in like 293s or whatever cell line because they're really easy to grow. They have like 1 million chromosomes. So they're just like, they're just really messed up, right? They're pretty easy to target. But if you wanted to practice or just see if the CRISPRs were working, you can transfect them into a cell line like HEK293Ts and look to see if the donor cassette is integrated by PCR or screen using a surveyor assay. And I've never done this, so I'm not going to talk about it, but again, they sort of discuss it on the CRISPR websites I told you about. So what I would recommend then is basically once you have your constructs and your donor or just your guide RNA is basically just to try using them in pluripotent stem cells. And the way that we do this in our group is to first transfect the cells. We've had the most success with nucleofection. So basically you harvest the cells as a single cell suspension, nucleofect them to get your plasmid or 2 plasmids into the cell. You then replate the cells and let them grow up for a little bit, and then you have the option of purifying the cells that got your plasmid using a number of methods. So I mentioned that there's plasmids that contain a GFP, there's plasmids that contain a pyrimycin, resistance cassette. So you can, what we commonly do is use the GFP plasmid, and then a couple days after nucleofection, you take your cells and you go to the cell sorter and you sort out just the green cells. And so you know that all of those guys have your Cas9 and your guide RNA because they're all green. And that really is going to obviously increase the likelihood that when you go through all the screening, you don't have to screen 100 clones to find one that actually got cut. At this stage, if you're doing homology-directed repair, so if you're using a donor template, you can screen for successful integration of the donor cassette. Again, using antibiotic selection if you have included an antibiotic resistance cassette. And then you can clonally expand the colonies, bless you, and screen them again for successful targeting. So again, this is kind of vague because it like really just depends on what you're doing. So for example, if you are using a plasmid that's going to insert a reporter, it's pretty easy when you design your donor to design a PCR screen, right? You know what the sequence was, you know what it should be when it's repaired. You can design primers that bind, for example, inside of your reporter construct. If you're trying to do something like make a single base pair mutation, you might have to design a more complicated screening process where, for example, in addition to introducing your change in the base pair of interest, you also introduce a couple of sites for restriction enzymes, so that if you PCR out that sequence, you can just do a restriction digest screening and see like if any clones seem promising, and then you can sequence it. If you're just doing no template, if you just want to see if there's indels, you basically just have to PCR out the sequence and sequence it. So again, basically the moral of the story is at the end of this, you're going to have to figure out which clones were targeted. And so when you're back at the beginning designing all the constructs and thinking about your strategy, make sure to think about the end when you need to figure out what got the right, what was targeted or what was cut or what edited properly, because that's really, really important. And it might be necessary to screen, 10, 20 colonies, so you might need to think about how is an efficient way to do this, for example, if you don't want to send all of that for sequencing. One more note is that in our group, even with antibiotic selection and sorting of the cells after nucleofection, we found that it's been really useful to do single cell sorting of the targeted line, which basically means that you bring to the cell sorter a plate coated with MEF feeders, and you sort one cell into like a 96 well plate, and you sort one cell into each well, so that when colonies grow up, you know that there's no mosaicism, because it can be a real problem if you have two cells that stick together, one was targeted and one wasn't, and now when you do your screening, you see a band. But of course, half the cells actually don't have it because PCR is a bulk screen. So it's really useful to know that your cells were basically very cleanly clonally derived. Right, so I basically went over this, but again, you want to think about the screening at the beginning. So for non-homologous end joining, you can do a surveyor assay. I feel like no one in our group has had particular success with this being a useful predictor of targeting. But you definitely want to sequence the region of interest. One note about this is if you're trying to do homology-directed repair with a template, and especially if it's not working, you can screen for non-homologous end joining just to see if you're cutting. So if your targeting isn't working, if you're trying to put a reporter in and it's not working, it might be that your guide RNA isn't cutting, and it might be that there's something wrong with your donor. So screening for non-homologous end joining can be a useful way to figure out like, are you getting cutting, but something's just going wrong with the homology-directed repair. And I mentioned this also, so for small edits like a single base pair change, you can design a donor template that introduces restriction sites so that you can do a restriction digest screen. For large edits, you can amplify using a primer that binds in your donor and one that binds outside. And you want to do one that binds completely outside of the arms of homology, so you're not screening just like a random site that got your entire donor. And that's basically it. So the general summary is the most important things are to design your guide RNAs well. It's hard to know whether your guide RNAs are going to work. One can seem that it's like the perfect guide RNA, it's right where it should be, it has no off-target effects, and it just doesn't work for some reason. And I think that the field is moving forward in terms of understanding that, but that's why I suggest picking at least 3. I know George in my group, for example, he designed 3, all based on like the same criteria, and tested all of them, and 1 worked and 2 just like did nothing. So it can be really helpful and save you a lot of time. You can then generate the reagents. Again, this is all pretty straightforward. I would recommend using the Zhang Labs CRISPR reagents and their protocols online, because it's the most straightforward and the cloning is really easy, and this should not be a hang-up. I would also recommend if you need to design a donor plasmid, hopefully you could talk to somebody who already had a similar plasmid that you could sort of just clone into, because obviously making that from total scratch is going to be a lot of work. Then you can, once you have all the constructs cloned, you can test them. Again, I recommend sort of skipping the normally recommended testing for iPS work, just because iPS are so particular and difficult to target that it's worth just investing the time in trying to target the line you actually want to work with, instead of spending a month targeting 293s. To do that, you can use whatever method is appropriate to get the plasmids into the pluripotent stem cell line, but we've had most success using nucleofection. And then finally, you have to clonally expand and screen. And again, I recommend single cell sorting if possible with whatever setup you have, just because it eliminates the possibility of mosaicism, or at least for the most part does, and then do a sort of thoughtful screen for successful targeting. Anyway, that's all. And then if people want to, I don't know, I know some of you are from a class, and I don't know if you care about the actual nitty-gritty, and then some of you from the course might want to look. I can connect, I think, using the Wi-Fi, and which might take a minute, and then show you on my computer if people have particular questions. Okay.
Can I ask a question?
Yeah.
Just practicalities of the actual nucleofection or inspection part. Obviously, as we're being taught to passage cells, it's all about keeping them in clumps, never have single cells, they won't stick, they won't grow.
Yeah.
So when you're actually nucleofecting these cells, are you doing so when they're growing in their colonies, or do you dissipate them first, and then afterwards, how on earth do you get them to grow when you
Yeah, so that's a really good question. And has been, basically hoping. No. So what we found works the best is if you have a cell line that's adapted to M-teaser, they should survive single cell passaging.
Okay.
So you would single cell dissociate them, nucleofect them.
So before you nucleofect them?
Yeah, so you single cell dissociate them.
Yep.
Put them in the nucleofector, and then you replate them. They'll look really bad. They'll be sort of sickly and spiky, and you have to use ROCK inhibitor for the first few days. But then eventually colonies will grow out, and the cells will sort of recover. And at this stage, you can either do the antibiotic selection, in which case you'll just have a few colonies, or you can sort them. For the single cell sorting, it's kind of counterintuitive, but we've had the most success sorting them back onto MEFs, so that they have friends to hang out with once they're sorted by themselves. And I don't think anyone in our group has successfully been able to sort them onto M-teaser culture. So we sort them back onto MEFs, grow them up, and then readapt them to M-teaser if they need to be. So the main thing that's important in particular with this, though, is with all the single cell dissociating and passaging and screening, you really, really, really need to rekaryotype your lines after you do this process, because the likelihood that it introduces a karyotypic abnormality is definitely a possibility.
Sorry, Katie, I
Yeah.
didn't understand. When are you replating all MEFs? After the first sorting for nucleofection? No.
You can do it then, or you can resort them again later. Yeah.
Okay. After the resorting is when it's better to see them off MEFs.
Yeah, because you're gonna sort them one cell into each well, and it's helpful if you do that in coated plates with MEFs.
You just sort into 96 well plates, do you?
Yep. So our sorter has, it has the capability to just put one cell in every well, and you can even practice. You can bring them whatever cells, and they can show you that they can put one cell in each well. Okay, so this, for example, is what I would do if I were trying to target SOX2, which is a gene I study. I would go to the NCBI website and just search under gene for the gene of interest, in this case SOX2. And again, all these instructions should be in the printout. And then you can go to the genomic regions, transcripts, and products section and click on FASTA. You can probably click on something else, but that's what I do. And then click send to file, and then have it download the full GenBank. So great, so that just downloaded on my computer. And then if I open this in Snap Gene Viewer, so now this is the SOX2 gene totally annotated by the NCBI database. So it tells me the full sequence, and then it shows me the coding region. And so this is really helpful, right? Now I know where all of the features I might be interested in are located. So if I want to, for example, target a reporter, sort of like the one I showed you, where I target it to the 3' UTR and replace the stop codon with a reporter, I would basically just highlight this sequence and copy it.
So is that your sort of range for targeting?
Yeah.
200 100 in each direction?
I would say 100, I would start with 100 in each direction. That's what we were originally told, is like as far as you want to go from the cut site that you're looking for. If you do that and there's like nothing, which I've never seen, then you could start expanding. But yeah, we've always been told 100 is like how far you possibly want to be. You can also think about, so this is like an example of a time when you could think about like what your donor construct is going to be like. So for example, I could choose to take only the region in the 3' UTR because I want a donor that's not going to get cut by my guide RNA. And so because the donor is already going to disrupt the 3' UTR, this would sort of solve that problem automatically. It would be really easy to exclude those sequences from the donor, whereas if I have a guide RNA that's in the coding sequence, it's going to be more annoying to design a donor that doesn't have the guide RNA in it.
Excuse me, Kate.
Yeah.
This is a Snap Gene Viewer?
Yes, Snap Gene Viewer.
That's as far as I got.
Yeah, so yeah, there's Snap Gene, which you have to pay for, but then Snap Gene Viewer is free. So it's pretty convenient for this. If you do a lot of cloning, Snap Gene is really useful, but it's definitely easy to use Snap Gene Viewer for this. So then if I go to that CRISPR website, and so I already pasted in this sequence before, I can just show you. So you can name it whatever you want. And then, so here's where you can select, for example, your genome assembly that you want it to use. And then you can also tell it what kind of Cas9 you're using. So again, this most common one is going to be this NGG motif. But if for whatever reason you need to use a different one or your group uses a different one, you can select different options. So then you submit that. When you do that, it comes to this page. And again, I'm not as familiar with this, but the general idea is that it ranks it based on the specificity of the guide to the sequence you're interested in. So it's basically trying to tell you which regions are like, there's a risk that the guide might like bind a random other gene and cut it. It will also tell you whether those regions that it also targets are in introns, are they intergenic, are they in an exon, and you might want to avoid ones with potential off-target effects, like in a coding region of another gene, just in case. So what I would usually do then with this sort of output is, basically the idea is you want to balance between the potential off-target effects and how close you are to the site you're trying to target. So I think this TGA is the stop codon. So these ones are all yellow, which means they have some number of off-target effects. And these ones are green, which means they're a little bit better. So I might, for example, pick like a couple of these ones that are a little bit farther from where I want to cut or like where I want the donor to target but are like better in terms of off-target effects, and then maybe pick like one of these that's like right on top of the stop codon but might have more off-target issues. The hope being that one of these guys is going to work just fine, and then I won't have to worry as much about off-target effects. But then if it is particularly difficult region to target, having one of these might also be useful. And again, that's kind of a personal preference in terms of where you, the balance between those two things. So say I wanted to pick this guide RNA. So I would just take this sequence, and copy it, and then you can go back to Snap Gene Viewer and just paste it, and then I can say, there's my guide RNA. So now when we look at the map, see I have my little guide RNA there, and now when I design my donor, I have all the information I need to know where this CRISPR is going to bind. And then once I have this sequence again, you basically just, I use the protocol for the BBS1 plasmids, so I would just take this sequence and order oligos containing this sequence with basically sticky ends, so that when you anneal them, they have these particular sticky overlays, and then you just cut the plasmid, stick in your oligo, and then your guide RNA is ready to go. And that's about how trivial it is. Yes, Greg.
So you talked about when you're trying to do a small insert, you would introduce some silent mutations so that you don't cut your donor.
Yeah.
How do you know that the mutations that you're putting in are going to be silent?
Yeah, that's a great question. So that again is when it's helpful to have this like totally annotated sequence. So using this, I can see the full coding sequence of the gene. And so say there was like a mutation here I wanted to change, and then add a couple silent mutations, I could know that, okay, whatever mutation I make, like this needs to stay a serine. And then basically just using like the codon sequences, you can say, okay, I can change this to a G and it will still be a serine, and it will also introduce this site. So you basically need to just sit down with like a pencil and paper and go through it and sort of think about the potential options. Think about like, okay, what region would I PCR out? Where could I introduce a cut that would be a potential screening site, and how can I do that without disrupting this sequence? And the main thing along those lines I would recommend is definitely make another file with like your end sequence, so like what it should look like if your donor gets inserted exactly as it is and everything, because then you can double check, right, and make sure that you didn't just like make a mistake and change an amino acid or something like that. And that's again where this is a pretty useful tool, because it will just like let you do all of this in silico cloning.
Can I ask you something? In the vector that you clone the guide RNA, there was the same restriction site, or there were two different sites?
Yeah, it's
the same.
It's the same site.
So you don't worry about multiple.
No, the whole point is that it's supposed to cut both of those sites. So it like cuts out a chunk, like a very short, they're really close to each other. So it basically cuts out a short chunk to have these like particular overlapping ends, and then when it anneals your guide RNA in, it deletes one of the sites. And then you can screen to see if you got your guide RNA inserted using restriction digest screening. No, it will only insert one just because of the way it's designed.
Okay.
Yeah. It's supposed to use both of the cuts. Yeah. It's a good question though. It took me a long time to figure out how it works. Yeah.
In your experience and that of, I guess, the whole institute, is there any problem with designing guides which overlap exon and intron kind of sites? I guess if you're looking right at the end of a gene, for example, you're going to go from exon to intron.
So
presumably it shouldn't matter.
right, yeah.
And it doesn't.
In my experience, so a lot of the guides I use target like right at the stop codon,
Yeah.
and it's been
And
like
it's fine.
completely fine, yeah. It can actually again be kind of useful, because then your donor, for example, is guaranteed to disrupt your guide RNA, because you'll only have half the guide in your coding region, and then the rest of it will be gone. Yeah, so that's a good question though.
So when you're designing the donor,
Uh-huh.
how big does the homologous region have to be on each end?
So for a plasmid donor, I usually try to make them around 700 base pairs on either side. Yeah. I don't know about the oligos. I think the oligos are pretty short. You've never used them, have you?
90 base.
Yeah, they're only like 100
150
total.
to 200 base pairs long in total. So the homology arms are about 70 to 90 base pairs.
And if anyone has particular questions about that, I can introduce you after to our resident oligo targeting expert, who I know is around. But yeah, 700 is good for a normal, like, large construct targeting.